Hello everyone, welcome back, and I hope you're ready for the upcoming session. In the coming hour, our distinguished scientists will talk about biological intelligence, how life sciences inspire new frontiers in data, diagnostics, and discovery. We're excited to have two laureates joining us to share in our panel discussion, and the session will be co-hosted by our very own local scientist, Professor Danny Chan, followed by a Q&A session. Professor Kazutoshi Mori and Professor Peter Walter were awarded Shaw Prize in the discipline of Life Science and Medicine in 2014, in recognition of their outstanding contribution in the discovery of the unfolded protein response of the endoplasmic reticulum, a cell signaling pathway that controls organelle homeostasis and quality of protein export in eukaryotic cells. Now, may we invite our speakers and moderator to this stage. Let's welcome them. We have Professor Kazutoshi Mori, Distinguished Professor, Kyoto University Institute for Advanced Study. Let's put your hands together to welcome him to the stage. Next, we have Professor Peter Walter, Professor, Biochemistry and Biophysics, University of California, San Francisco. And hosting the session for us, we have Professor Danny Chan, Emeritus Professor, School of Biomedical Sciences and Director of Development and Education of Research Integrity, the University of Hong Kong. Welcome again. Please take your seats. Now we're ready to be inspired. Over to you, Professor Chan.
Okay. Hello, good morning, and welcome to this session, plenary discussion. Today we have two very distinguished laureates with us, Professor Peter Walter and also Professor Kazu Mori, who will discuss with us on the topic of biological intelligence and the role of and the role of how life science inspire new foundation in data, diagnostic, and discovery. From the perspective of the two laureates. And I'm also super excited and honored to have the opportunity to chair this discussion session. Now, biological intelligence is a huge topic, which will probably take hours to discuss. But maybe perhaps we should first touch base on the later discussion. And we will first focus a little bit on how life science inspire new frontier in data, diagnostic, and discovery. Perhaps from the two laureates, Peter and Kazu, if I can address you by your first name, to keep this forum a little bit informal, as we all already dress quite formally as basic scientists. Yeah. Now, yesterday, I think Peter and Kazu gave two really wonderful lectures on the mechanistic insights and perhaps potential translation opportunities from their favorite topic of the unfolded protein response, or the UPR, from which they have both won and share many prizes and counting. And of course, the Shaw Prize. Now, on how life science inspire new frontier, Peter and Kazu, I am sure the audience would love to hear a little bit from you, a bit from the historical background leading to your discoveries. So my first question to both of you is what made you interested in cell biology in the first instance, And what make you, the decision making in choosing this topic to focus on the unfolded protein response, yeah? And perhaps maybe a little bit on your discovery process, whether it is a targeted discovery or whether it's something serendipity. Yeah. So maybe perhaps I begin with you, Peter. You could share a bit of your early career research and leading to that discovery.
Yeah, so I grew up in Berlin, in Germany. And then I started studying chemistry. I went to the United States at Vanderbilt University, get a master's in organic chemistry. And from there, I went to Rockefeller University, where I got my PhD with Günter Blobel in serology. And what impressed me when I first visited as a totally naive chemist, entering the laboratory of serology, there were these beautiful pictures, electron micrographs of George Pallade on the wall, of how beautiful and how complicated cells are. And I think that sort of motivated my transition from chemistry, which is pretty much more an engineering discipline. We make molecules, we synthesize them, to biology, where we are just faced with the products of evolution, and we are trying to make sense out of them. So it is a path of continuous detective work. And since evolution takes pieces that are lying around and puts them together in different ways to make them useful for a new function, these are Rube Goldberg machines, where the rules are not really deterministic of what is being used for what. So that the spectrum of discoveries you can make is absolutely enormous. So I worked in Günter's lab on protein translocation into the endoplasmic reticulum. I discovered one of the components based on my graduate work, that is the signal recognition particle, and characterized its function, basically biochemical. And with that accomplishment, I got offered a faculty position at UCSF, where I then started my own lab and continued my career for 40 years. And in this course of transitioning to a new place, I was exposed to a new environment, new mentors, senior faculty members, and one of them was Ira Herskowitz, and Ira was a yeast geneticist. I took his genetics course as a young assistant professor. I took a class with him and learned genetics. And now my lab was ready and was eager and wanted to do some genetic experiment. So the unfolded protein response came out of the desire of two graduate students in my lab who wanted to do something genetic in the lab. And the question we came up with is that proteins, as they enter the endoplasmic reticulum, which we were the experts at the time, now they have to fold up. So how do they know, how does a cell know that it has enough folding machinery to do so? There was previous work, some from Kazu's work, that the transcription machinery in the nucleus provides the proper amount of chaperones. So it makes more mRNA for chaperones, they enter the endoplasmic reticulum. So somehow the endoplasmic reticulum had to tell the nuclear compartment what it needed. There needed to be a feedback loop that establishes the appropriate amount of folding machinery for the amount of proteins that have to be folded. So this homeostatic event cannot happen just randomly, so there must be communication, and we set out a genetic screen to identify components on that communication pathway. And that screen led to IRE1, which then turned out to be one of the most marvelous and beautiful enzymes. We are still working on it today. There's going to be a poster by San Wen Hu in the lab, my lab currently, who will present this tomorrow. It's one surprise after the other, and we keep working on it. And it's a single transduction device that sits in the membrane of the endoplasmic reticulum, figures out with one end what's going on in there. Do we have unfolded proteins? Do we need more chaperones? And then it transmits that information across the membrane into the cytoplasmic space, from where it then activates the transcription machinery that provides the appropriate gene products to make more of the chaperones to take care of the proteins. So it's a very beautiful little loop of signaling, and it uses very unusual machinery and bits and pieces that have kept us busy for literally decades and have produced many PhDs.
Thank you, Peter. Ka-shing, perhaps you can share your perspective?
Nearly 50 years ago, I was a freshman of Kyoto University. At that time, I did not like biology. Textbooks seemed very too descriptive. So I was a student at the Department of Chemistry, a faculty of engineering. But when I was reading newspaper, I found the interesting term, molecular biology. What is it? I searched many articles by myself and found that idea of central dogma. DNA carries genetic information, it's transcribed to RNA and translated to protein. And this genetic code is conserved from E. coli to human. I was amazed that biology has a beautiful, simple theory. So I wanted to learn molecular biology. So I changed my major from chemistry to biology, just like Peter. But molecular biology is the most advanced science, so not so many laboratories can work on it in Japan. So I chose biochemistry. I was biochemistry nearly 10 years and got the position of assistant professor in a local university in Japan. I kept working on the biochemistry, but I could not stop my passion to the molecular biology. So at age 30, I decided to go to the United States to learn molecular biology. So my boss at University of Texas Southwestern Medical Center, Joe Sambrook and Mary J. Gasing, accepted me as a postdoc in 1989. 1 year before, in 1988, they published a paper in Nature that showing that our eukaryotic cells are equipped with an interesting system called UPR. So that's my start. So when I become a postdoc in their laboratory, UPR system exists, but no molecule identified at all. So I was quite fortunate. I could start from beginning. Then it's a transcriptional induction system. I analyzed promoter region of chaperones and discovered cis-acting element responsible for the transcriptional induction we named UPRE, unfolded protein response element. Then published a paper in EMBO Journal, 1992. Then usually, next step is purification protein capable of binding to UPRE element. But I was a biochemist. Purification protein means hard work in a very cold room for a longer period. I didn't repeat biochemistry again, so I wanted to challenge new things in United States. So I challenged yeast genetics. I isolated a mutant which unable to induce chaperone even in the, under the condition of ER stress. And I screened 100,000 colonies and worked for half a year. I got 3 mutant cells. Then it took another half year to get gene that defective in that mutant. Then I got IR1, or Peter said, we had that same molecule. I had IR1 as an ER stress sensor. Then It's published in 1993. Since still I'm working on this UPL system, I switched from E-cell to mammalian system, and now we're working on the mammalian UPL, or using fish model, still working on the UPL system, biology and physiology of the UPL.
Thank you, Peter and Ka-shing. So having discovered the very first sense of the UPL obviously is very important and has opened up a huge area of research for yourself, I guess not only for yourself, but also for many other researchers like myself as well too. So I wonder if you could share with us, how you take your initial discovery and to further, to take on that challenge to look for more information and how you would inspire from that. Maybe I'll start with you again first, Peter.
Sure. So the first screen was done by 2 graduate students in the lab, Caroline Shambu and Jeff Cox. They collaborated wonderfully for the screen. And once we had the first component, then the discussions began. Now, what shall I do and what shall I do? How do we divide our work so that the absolute connectivity of doing a screen together didn't last any longer. So we had to somehow divide the project to give them individual freedom to develop their own path forward. So Caroline chose to be more involved in characterizing the molecule we had already identified. And Jeff said, well, I'll just do another screen and find the next component downstream. Because once we have this signal transduction device sitting in the membrane, it has to talk to something that then talks to the transcription factor. So we did another screen and we pulled out the transcription factor that happens to be activated by IA1. It's activated in the most fascinating way by having a very unusual splicing reaction. So the field started exploding we exploded in different directions. We stayed in yeast for the longest time and determined the mechanism, which then beautifully, most of the salient features held true in the higher metazoan cells and in mammalian cells and in fish. So it became it spread out into many avenues that became complementary to one another.
So after publication of IRE paper in Cell in 1993, I went back to Japan and I got a position as a researcher in an institute which existed only for 7 years contract. Then next target is transcription factor. Then, again, I did not want to purify a protein, so I managed to try to identify factor by molecular biological techniques, but it's difficult. I struggled for a while, but one day I came up with the idea of one hybrid screening. And then this idea, one hybrid screening is a modified version of 2 hybrid screening, which works only for transcription factor. Then this screening works well, and I got HAC1, like Peter had Simon. Then find the splicing of HAC1 mRNA. But one hybrid, as I mentioned, one hybrid screening is a modified version of 2 hybrid screening that can be applied to any other species. Then using the same strategy, we could get mammalian transcription factor after identification of 6 acting element in a mammalian chaperone promoter, which explain simultaneous induction of many chaperones. So now then that identified 86 BPN, 2 key players in the mammalian UPL.
Ka-shing, perhaps I'll ask you a further question. Because having spent time in the US, you decided to move back to Kyoto, right? And you think that it was the right decision. It was a very important decision to move back to Kyoto. So you had some premonition that U.S. going to get into trouble, or is there a particular reason why you wanted to move back to Japan to carry on your career?
So when I was working on the IRE at the last stages, my boss Joe Sandberg asked me, "What do you want to do after publication of this paper? If you want to stay in the United States, I can give you time and place for job interviews. But if I keep staying on UT Southwestern, it's easy, and I will happy to do that. But to get the position, I have to go somewhere else, an unknown place, and create a new relationship with new faculty members. I thought it's kind of a little bit difficult. Then I decided to go back to Japan. But I found a position in the institute. It's well funded. The new boss, Dr. Yura, is a gentleman, but when I want to isolate transcription factor by multi-copy suppressor screening, but he said no. Only that time he said critical, but no, because IRE is a kinase. So if we use multi-copy suppressor screening, there might be more kinase. You may get kinase, but never reach transcription factor. You have to find a method by which you can directly get transcription factor. It's a difficult job, but I managed to came up with the idea of one hybrid screening. Then that kept me going. So that was very good advice.
Thank you. So Peter and Ka-shing, since that discovery, you both have published extremely well and continue to discover the mechanistic insights into the process. And you two have become really good friends as well, right? Research these days, research is supposed to be fun, right? Full of curiosity. But many young investigators these days actually feel research to be highly competitive and a little bit stressed out as well. So was this also an issue for you? Or it was, you consider it as a synergistic discovery path?
Well, shall I start?
Yes, please, Peter.
I think it was fun competition.
Peter was a big war to me.
We were definitely competing with each other, and I think it was absolutely productive and fantastic in the end, because we were all out. We were making many serendipitous discoveries here that were not the usual mechanism that happens. We have a very unusual splicing event in mRNA for transcription factor that IR1 is initiating, and so on and so forth. So pretty much everything we found, or what Kazu found, we wanted to prove wrong in the respective labs. And then we corrected each other at some point in the past, and we confirmed each other in other ways. And by having this sort of dichotomy between the two different groups, trying to do the same thing, but never publishing a paper together, everything was, even no matter how strange the mechanism seemed at the time, it was accepted by the field very quickly as being, yes, this is the way it is. So science moved much faster, much more directional. We didn't bark up the wrong trees for too long. And I think it turned out to be absolutely a fantastic relationship, even so there were a few painful moments, but it was overall great. So don't shy away from competition. Just make it friendly, right? It's not personal. There's nothing personal in it. You don't start hating people. You just go for what their science contributes, and you argue on with data.
Ka-shing, would you like that?
in 1990s, Peter was a big wall. So I have to keep working on hard. And when journal Cell come to our institute, I checked all index. Oh, I don't see Peter's paper. Okay, then I read papers carefully. But Peter said that this competition derived this field dramatically forward. And then I could move on to the mammalian system because ECUPL is too competitive for me. I find a way to go by myself.
Right, so the message to the young people is that networking is very important, collaboration is very important. Don't keep information to yourself, share it. I think this is the way to sort of, work together synergistically, just like what Peter and Kazu do. Right. Okay, now it is actually well recognized that there are many diseases associated with the protein folding and cellular stress pathways, right? So given, so I guess a key question is that how your discovery in cell biology have influenced understanding of disease and innovation in diagnostics and potential therapeutic outcomes. And maybe any clinical breakthrough that you may like to share with us, from your own perspective or even from your own research. Maybe Peter first, and then Kazu, if you want to add to that.
Yes, so we started out, me being a chemist, slithering into cell biology, we started out with very basic foundational research. Trying to figure out mechanistic details. And it eventually became clear as the field emerged from our work, from many other works, that protein folding and the whole idea of quality control in the cell is tremendously important for many disease settings. And then mostly through collaborative work, we started engaging with groups to apply our knowledge to see what we can do about these different diseases. Cancer comes to mind. Cancer cells evolve in your body in a certain spot. They become ever more genomically unstable, so they make many mistakes in the proteins they produce. That turns on these surveillance pathways. Not just the unfolded protein response, but also heat shock response and others. And so the general image emerged here, right? That as we fix these stress pathways, as we modulate them, learn more ways where we can potentially drug them, we could do good things. Because in cancer cells now, these pathways are activated. They make decisions, life-death decisions for the cell. Most of us don't have cancer because the cells that have these abnormalities are programmed to die. And if these events go wrong, because the unfolded protein response does not make that decision correctly, then we get cancer. So the cancer cell then uses these pathways as a band-aid to cover up, to have enough chaperones, to cover up all these passenger mutation, proteins that have these passenger mutations that are otherwise seen as unfolded proteins and as detrimental. So if we take this away from them, we can selectively kill cancer cells. So we engaged by now, it's a over 10-year long collaboration with Avi Ashkenazi at Genentech, who is developing new ways of chemotherapy based on blocking the unfolded protein response, and is now in preclinical development and looks very promising. It's very non-toxic because these stress pathways are only activated in cells that are unhappy, that have stress, that have misfolded proteins. So we can select these drugs and these inhibitions pretty targeted to the diseased cell and eliminate it from the body. So that works great. And then as the field of protein quality control emerged, the other branches of the unfolded protein response emerged, the PERC branch and the ATF6 branch. We found very similar opportunities in these branches. So blocking the integrated stress response branch that is mediated by PERC, among others, we got into neurodegenerative disease, and that's what I talked about yesterday, showing a little small molecule that we isolated that shows a great promise of fixing various cognitive defects in a numerous set of diseases. So again, we are approaching it disease agnostically at the beginning, trying to figure out what are the mechanisms by which things work. That gives us handles of surgically interfering with these pathways. And then through collaborative efforts of people who are having these disease models in their labs, we can advance it into potential therapeutics in the future. And again, this small molecule that we discovered in the lab is now in clinical trials for a variety of cognitive disorders.
Yes, medical doctors are quite busy by taking care of many patients. So medical doctors do not touch area field with mechanisms unknown. But our basic researcher investigated a system and clarified the mechanism. So by doing that, we can show marker for ER stress, splicing of XBP on mRNA, or phosphorylation eukaryotic translation factor, or just show the marker. Then medical doctor checked this marker is up, no, in disease they are looking for. So then many papers come out showing relationship with disease and ER stress. So basic research is really important to open up new diagnosis, treatment of diseases.
Thank you. I think Peter, yesterday talk on ESRAP was fascinating. This actually makes me think about what I should take some of these ESRAPs to improve my cognitive function. It's super amazing. So we know that a cell actually needs to make many decisions, right? And none more so than to live or to die. And the UPR, in a way, is actually central to such a decision. So given the topic of the panel discussions is on biological intelligence, I was wondering whether perhaps I can get your view on your thoughts on biological intelligence, and can a cellular decision making can be considered to be intelligent per se, or is this to be at a much higher level of an organism? So can I get your thoughts on this? Maybe again, Peter, start with you, and then Kazu, if you have anything to add.
Yeah, I was sort of surprised by the term the intelligent cell being used. So as I said, I trained in chemistry, so for me, a cell is a collection of molecules that all obey the rules of thermodynamics, and as such, their behavior is deterministic depending on the concentration of things we have and who is seeing each other in the right place at the right time. So I would say it's really in the language here. What do we consider intelligent? So in my view, a cell is not intelligent. A cell makes appropriate decisions depending on what the programs are, the molecular programs that it carries out. And these decisions have stochastic elements in them. So the cell death is always a fraction of what's happening because of the molecules that are available and that are doing these things. So the decision in the end is pretty deterministic in the population, and and this is evolved in a way that the ribosome is just good enough to make proteins with high enough speed and high enough fidelity. It doesn't need to use intelligence, right, to decide whether it should abort protein synthesis or not. That just happens because the way the molecular components interact with one another. So I think the term is overused, and we should leave real intelligence, which for me involves making a creative solution, right? That is, and I haven't seen it. I haven't seen a cancer cell making a creative solution to become better.
So it's more of an existential decision rather than...
Yeah.
Ka-shing, do you have anything to add to that?
I asked my colleague, please ask AI how cell decide survival, death under ER stress condition. Answer is depends on duration and strength of ER stress. If duration is become longer and strength become heavy, cell decide to die. But it's like no decision, like just heavy ER stress overwhelming cell, and they have to die. But I don't. There may be a switch, real switch from survival to death. So if there is a switch, that might be intelligent. So we are looking for such a switch. No mechanism is not always activate from the beginning. Not activation in the data time or cell decision time. So still we need to find a switch, I think.
Okay, so I do agree that intelligence is kind of overused, in particular in artificial intelligence. We touched a bit on this in this morning's discussions as well too. But certainly, AI is having a huge impact in our daily life, and the same in scientific research as well too. Because many of my colleagues now actually are using many tools they call AI. None so in protein biochemistry in terms of protein folding, for example, AlphaFold, AlphaFold 3 and so forth. But that is really leveraged on, decades and decades of really hard work, right? Looking at the structures and so forth before we actually come to that. So really, in terms of, it's leveraging on information rather than artificial intelligence per se, it's using information that we have to produce information. Now, a dream of cell biology like myself is really to develop an artificial cell. The dream is to try to understand every component in the cell, every molecular interaction, every assembly, and so forth, and such that we have a digital cell, and we can perturb certain things within that and see, with a specific perturbation, we'll now be able to understand what the outcome would be. My question to you is that, do we have such information? Is it possible? Do we have enough information to arrive at such an artificial or digital cell? If not, what more do we need to get? Again, Peter, maybe begin with you.
Well, I think we're getting there in terms of information that we have now opportunity with new methodology to get huge amount of information, more than we can handle with a human brain. Wolfgang's talk yesterday, when you look at the tomograms, they basically contain the information where every component in the cell is. We just cannot distinguish them yet, unless they're big and like the proteasomes or the ribosomes stand out, and we know what we are looking at. So we will get there eventually. The problem with the data out there, on which AI relies, is of course that, and I think was said this morning and was said yesterday by Professor Mack, that probably 95% of the data out there are crap. And AI will not be able to distinguish what are the crappy data from the good data, right? And there are many, there's this gray zone in between, that things being done in different cell types may lead to opposite results because of some very trivial explanation what makes these cells different from one another. And all of that needs to be integrated at some point. So I think we are far from being able to build a virtual cell. We will be able to build virtual models that allow us to have a coarse-grained understanding. If we tickle this receptor, this will happen in terms of transcriptional program. And if we have this transcriptional program, this will happen. But it will take a long time to really be able to then zoom down and say, these are the molecular events that happen. These are all the components that are involved in that. Because one little thing, right, one little phosphatase that takes a phosphate off that we don't know about can change the picture entirely. And so we need to do both, right? We need to keep on creating data. We need to make sure that the data we produce is accurately described, so what the weaknesses are, what the strengths are, what the cell types are, what the time points are, and so on and so forth. All the conditions, all the stuff that's now shoved into supplementary material and becomes so important for the condition will become more and more important because now AI will actually read that stuff and bring it back and allow us to integrate it into our models. So it's a tremendous tool of doing all the reading for us and the integration. But we have, whatever comes out of it, we have to see with a grain of salt and put it to the experimental test.
Do you have time to add to that?
Yeah, AI really helps researchers. AlphaFold can predict three-dimensional structure, but now new AlphaFold can show protein-protein interaction. It was very helpful for our story moving forward, but I have two concerns on the AI direction. Because one is, Peter said, there are many bad papers. And how AI can discriminate good paper from bad papers. So if you rely on the bad paper, the direction going wrong. And second is experimental condition is so diverse in biology. Many try and error is required to find a good way to go. But AI can do, and then during such try and error, there may be jump for a good idea. So AI can do or not, I'm a little skeptical.
Thank you. So obviously, I think we probably must use AI as a tool, but don't over-rely on AI. We must understand what is true and what is rubbish, right? So we still need to think that from that perspective. I know time is up. I would like to close up with one last question. We now know that a cell can get stressed out and can sense it and make existential decisions. Unfortunately, I think the changing in research culture and now politics and the competitive nature of research also places a huge pressure on many, if not all, the junior researchers and scientists. And they are all stressed out quite a lot, okay? So I wonder if you could provide some advice for the budding young researcher out there, how to actually best approach this ever-changing research environment and demand from our senior management and regulators. Final comments, please.
Well, think what the cell would do, right? It will turn on the stress pathways, and the stress pathways will try to bring things back into balance or kill the cell. You don't want to kill yourself. So you only have one option. Bring your life into balance, right? And see things in perspective. I had graduate students who were working incredibly hard and who were going nowhere with their projects because they didn't take the time to think. They didn't take the time to think of what was the important next thing to do. They just kept on doing, like hamsters running in a wheel, going nowhere. Don't put yourself in that position ever. The other thing is you need to realize that the profession we are in, 95% of what we do fails. And that is good, because if more things succeed, then you're only doing the obvious. So you want to challenge yourself and be at that level of risk-taking. So make sure that your life provides a balance to that, a hobby, do some sports, and just bring your life into harmony, and the stress will go away.
My advice is, I talked yesterday, find your own strategy to make progress. It's really important. Getting a good strategy change your life. So I hope you can find your own way to go.
Well, thank you. Thank you, Peter and Kazu, for this advice. I guess, I think a balanced work-lifestyle is very important. 7/24 in the lab doesn't necessarily bring you what you want. Time to think is more important, right? I think that the classic thing is think before you do, right? Now I think
but sometimes you need to be there 7:20.
Yes, yes, indeed.
Just use it selectively.
Yes, indeed. I think what I'd like to do now is actually open a Q&A session to the floor. I'm sure there's lots of eager junior scientists out there who would like to ask questions to both Peter and Kazu, so please. I think the lady with the mask got her first hand up, so please.
Thank you very much for the wonderful panel discussion. I'm Rafaela, so once again, and I have something to follow up regarding the limitations of AI, especially in terms of studying protein biochemistry. Just because from my own research on Parkinson's disease, there's so much stochasticity, and just with, as you said earlier, like random cellular environments influencing protein misfolding. And in my own research, actually, even if you keep everything the same, just random proteins doing random things themselves could cause completely different results. And so my question to you is, because experimental science will continue to be so important in the context that AI cannot fully predict these things, what are some future kind of techniques in terms of bench science for protein biochemistry that you predict will be very useful for expanding the field of trying to understand protein misfolding, protein unfolded response, stuff like that? Thank you.
Yeah, I think a good angle in which to think here is that most data in the structural protein database are still derived from crystallography, right? We are now going into a predictive mode from that through AlphaFold. But we are also having new techniques with single particle cryo-EM, which gives a more native state of protein in solution. And what we are learning there is that proteins are not locked into a certain conformation. If you do molecular dynamics on a kinase, it looks alive, right? The surface is fluid. It constantly wiggles, and so every protein we have is an ensemble of states, of many, many different states that are interconverting. Some of them are good, some of them are bad, some of them just happen to be there. So by gaining more and more appreciation for that fact, I think a lot will come out of applying molecular dynamics and exploring this randomness, right? When do you fall into a trough? When are you stuck in one reaction or another? And what influences that? There's the stochastic element, and then there's the butterfly effect, right? That in climate prediction, that a tiny little change here can have a huge change here if you just change the trajectory. So as we're gaining more understand and appreciation for the fact that things are kinetically controlled, and to a large degree a statistic with many, many, many decisions that are constantly being made, or different paths being chosen. We're going to get a better appreciation for what we are up against.
Okay. Another question? Could I ask you to actually keep the question short and concise? Yeah? Please, lady at the back got her hand up.
Yes, my question is, did you have any apprehensions when you were about to switch from chemistry to biology? And would you recommend for young scientists to expand their research area during their early careers?
Young scientists probably needs good mentor first. To find an interesting field. If you are interested in biology, then you don't hesitate to change major to something from something to biology. Then find a good mentor. Then a good training, and then determine what to do after getting PhD. It's most important thing after PhD is find a direction, which field you wanna go. So that's stepwise fun. Find a good major, and find a good mentor, and good education, and find a way to go after getting PhD. So my advice is that answer to your question.
I think it's one of the privileges we have in our careers, right? That we can reinvent ourselves, and that we can constantly learn into new things, right? Yes, I had apprehensions. At the beginning, you feel like a fool because you don't understand anything, right? You have to learn the lingo, you have to, I mean, you're in a totally new field, and this is, it's risk-taking, but it's also tremendously rewarding as your appreciation of the complexity of biology increases.
Thank you. We have quite a few questions online, and I'd like to actually pick one of them for you guys to tackle. How do you think the emphasis on translational research affects basic science? Is there a risk of losing curiosity-driven exploration?
In biology, curiosity-based research leads to some clinical success in the many years later. But so find basic research is important for new development.
Peter?
I mean, the basic research provides the foundation for the translational arm of clinical medicine. CRISPR came out of bacterial immune system, right? Telomerase came out of slime mold, the discoveries. So this is something that our funding agencies need to justify what they've done with the money. They need to provide more, bring in more money by in the United States, providing Congress with nice examples of how health has been improved. So I think basic science, basic discovery, foundational research is constantly endangered because it's seen as here's a scientist playing around and not knowing where they go, and not really mission-oriented. So it is constant struggle to provide the examples of where has basic research been the important piece in a translational translational accomplishment. So that's why we're learning all this lingo. When you write a grant, right, you need to write down where this is going to go, which is bullshit. Because for all the things that we have done, we had no idea where this would go, and this only came when we had the data. So somehow, I'm not sure how we get out of this devil's cycle that we're currently in. So yes, there is a danger.
Yes, thank you. I guess there needs to be a balance. I think, innovation in technology and focus on translation is important so that we can translate what we discover in the lab. So we have senior management here, and why I think we should push for translational innovation. Let's not forget curiosity, basic research that has driven many of the important discoveries in the past. I'm sure it will be so for the future as well too. So back to, yes, the lady's had her hands up for quite a few times, so please, the lady at the back.
So hello, both professors. My name is Irina, and I work on dementia prevention as a therapist. So Professor Mori, you mentioned that you find your strategy to make progress. So can you share a little bit more about what your strategy is? And throughout the process, is this strategy developed before you start, or it's more like in trial and error? Thank you.
I came up with the idea of one hybrid screen to identify transition factor I'm looking for, but nothing produced anything. So I thought I came up with the idea by myself. I thought I'm a genius, but that in the United States, we had no journal club in the Joe Samuel club. So we didn't have enough time to read a paper. So we, Japanese scientists, get together once a month and show the new paper, new interesting paper. And then one guy showed one hybrid screen in that discussion. At that time, I didn't know how important it is, but some input in my brain. Then after coming back to Japan and struggling for why to find a way to identify factor, then that idea come down to me. So you have to read many papers. It's better to read many papers to get idea. So you cannot produce nothing from everything. I think it's by reading many papers, you have to train your brain. It's my advice.
And talk to many people. That's often faster and more fun, certainly, when you do it in the bar.
I understand the time. Maybe perhaps we have one last question online that may be relevant on the issue of intelligence. So the question is, what role do you see AI playing in hypothesis generation, not just data analysis?
I think it's a powerful tool. The AI will not generate the hypothesis, but you will generate the hypothesis in an AI-assisted way. When AI digs through large data sets, right, and provides associations that we were not aware of before, we can build them into hypotheses that we then can test. Right? There's enormous data in now that there is huge data banks becoming available for human mutations. Right? I mean, we all carry so many mutations. And the statistical links that are being made to phenotypes are becoming stronger, and sometimes you find that there are associations with something that may be worthwhile testing. So you build a hypothesis on that. You ask the programs to dig through these databases and see whether something correlates with your phenotype of interest. And then you have to do the experiments and see if it works. So yes, I think it can help tremendously when used cautiously. You don't ask it to put out a hypothesis. You just ask more subtle questions that then lead you to propose what is in your range of interest.
Okay. But I guess at the end of the day, you still really look at those, how those hypotheses are derived from the data that are out there, right? So even though the AI has put forward a hypothesis for you, you also need to really check and double check yourself that the data is accurate, right? Okay.
Absolutely.
Sorry, go ahead, Peter.
I was just agreeing with you.
Okay, right. So I think time's up. Unfortunately, I know that many of you still got many questions. I have to bring this session to a close. May I ask you to join me to thank both Peter and Kazu for this wonderful panel discussion. Thank you.
Thank you. Thank you very much to our panel for this wonderful discussion. Please remain on stage. We would like to have a photo of the three of you. We want to gather at the front of the stage. That was truly inspiring sharing from our three professors. During today's program, in the morning session at least, we heard several times that artificial intelligence was being brought up. The rise of AI is changing the way we live and work, but it's very important for us to be cautious about how we utilize it. Thank you so much once again, and thank you, Professor Chan, for moderating the session for us as well. Let's put our hands together once again to thank this panel for enlightening us. Thank you. The insights shared by our scientists this morning have shed light on the deep connections between life sciences and advancements in data and diagnostics. Thank you to our participants as well for your active participation. Let's carry forward the ideas shared today into your future conversations and collaborations. Thank you for joining us this morning.